Academy. Today we are going to talk about one of the important topic in the food industry, that is fermentation technology. Right? So as we know, fermentation technology, it is an ancient food processing technology and it has been used for the production of beverages, processing of meat and fish, production of condiments, staple food production, production for organic solvents, and also in the pharmaceutical industries. So fermentation, it is very important because it is being used in diverse industries, right? But if we talk about the definition of fermentation, it is a process in which chemical changes occur in an organic substrate through the enzymes produced by microorganisms and the important conditions in which this reaction occur is under anaerobic conditions, which means during the fermentation we do not need oxygen. So this process, it actually occurs in the absence of oxygen, right? For example, sugars, starches, and proteins. In the absence of oxygen, they all are fermented into alcohol and simple peptides or in amino acids, right? So during this process, another thing happens, which is the ATP production. This process is actually needed for the production of energy in the form of ATP, and this one is occur by converting the glucose molecules which are produced during the glycolysis pathway, and glucose here, it forms the NADH molecule with pyruvate, right? So during the fermentation, this pyruvate molecule is converted into variety of compounds, and NADH is actually regenerated into the NAD plus molecule so that it can again participate in the glycolysis and can produce more and more ATPs, which is our main focus, right? So as I said, it produces different type of compounds. So this one actually depends on type of fermentation, which is occurring under the anaerobic conditions, right? So now let's talk about the fermenting microbes, the microorganisms which actually are responsible for the fermentation. So if we talk about bacteria, so in case of bacteria, Lev species is the one which is responsible, like Enterococcus bacteria, right? Streptococcus, Leukostock, Lactobacillus, and Pediococcus bacteria. But in the fermentation, yeast and mold are also important. They also participate in the fermentation, right? So for example, if we talk about yeast, the microorganisms are, D. Baromyces, Fluoromyces, Geotrichum, Penicillium, Penicillium, and Rhizopus species. Rhizopus species. So they all microorganisms are actually used in the fermentation technology, right? So if we talk about the basic principle of the fermentation, so the basic principle of the fermentation is that to derive energy from carbohydrates, from carbohydrates in the absence of oxygen, right? As I said again, in this case, glucose is converted into pyruvate by simple mechanism which is known as glycolysis, right? So when there is no oxygen, then this pyruvate is actually reduced into alcohol or acid with the regeneration of NAD plus molecule. And then this NAD plus molecule is again participate in the glycolysis, where it again produces the more and more ATP as an energy source, right? So if we talk about in case of fermentation, only 5% energy is generated as compared to aerobic conditions, right? Now let's talk about the important thing about the fermentation technology. It is type of fermentation. Type of fermentation. So number one type of fermentation is known as lactic acid Homo fermentation. And in the lactic acid Homo fermentation, glucose is converted into lactic acid only, right? And the microbes which are responsible for the lactic acid Homo fermentation are Lactococcus, Enterococcus, Streptococcus, and Pediococcus. And if we talk about Lactococcus, then they are used in the dairy industry as starter culture for the production of different type of milk products, right? Second type of fermentation is lactic acid heterofermentation. Lactic acid heterofermentation. In case of lactic heterofermentation, what happens? Glucose is converted into lactic acid with the production of some byproducts. For example, acetic acid, ethyl alcohol, carbon dioxide, and water. And the microbes which are responsible for the lactic acid heterofermentation are leuconostoc, enococcus, coccus, vicella, and heterofermenting heterofermentive lactobacilli. They are responsible for the lactic acid heterofermentation. Another type of fermentation is known as propionic acid fermentation. In this fermentation, glucose again is converted into lactic acid plus propionic acid plus acetic acid and CO2 and water. Right? And the microbes which are responsible for this fermentation are propionobacterium. Propionobacterium. And some species of Clostridium propionicum. Propionicum species. Right? So propionobacterium, it can use glucose and lactic acid as their initial substrate. Right? For example, in sugars, if there are sugars are present in the environment, so in that case they use glycolysis pathway. Glycolysis pathway, which actually produces the pyruvate, and then this pyruvate then gets carboxylated into oxaloacetate. Right? And then this oxaloacetate, it gets reduced into propionate via malate, fumarate and succinate. So if propionic acid has to be produced, then it will go these steps, in which pyruvate is converted into oxaloacetate and oxaloacetate is then further reduced to propionate. Right? Another type of fermentation is diacetyl and 2,3-butylene glycol fermentation. In this case, two types of products are formed, and the substrate here is citric acid, which produces pyruvate plus acetyl methyl carbon. Then this acetyl methyl carbon, it is converted into either 2,3-butylene glycol or converted into diacetyl. And the microbes which are responsible for this type of fermentation are Enterococcus, Erwinia, Hafnia, Serratia and Klebsiella. Another type of fermentation is known as alcoholic fermentation. Alcoholic fermentation. In this case, glucose is converted into ethyl alcohol. And this one is the best known fermentation used in the food industry and carried out by either yeast, fungi, or bacteria. So in this case again, from the glucose, pyruvate is produced and NAD plus is regenerated again, which again participate in the glycolysis to produce more and more ATP, right? And it is done by reducing the NAD plus. It is regenerated by reducing the acetaldehyde to ethanol. So if we again write this equation, then it will become from the glucose, pyruvate is produced, right? And from the pyruvate, NAD plus is regenerated with acetaldehyde production. Acetaldehyde production and acetaldehyde then reduced further for the production of ethanol. That is why this one is called alcoholic fermentation. Another type fermentation is known as butyric acid fermentation. Butyric acid fermentation. In this case, glucose is converted into acetic acid and butyric acid. This one is carried out by the obligate anaerobic bacteria such as Clostridium species. Right? So in this case, butyric acid fermentation, if we look out for the whole process, then glucose is actually converted into first pyruvate, and then pyruvate is converted into acetyl-CoA, and it also produces carbon dioxide and water. And then this acetyl-CoA is used for the production of acetic acid. So this one is also called acetone या फिर acetic acid fermentation. Right? So these are the important type of fermentation in which we had seen 6 types. So if we look into the picture to make it more clear, the fermentation technology, then what's happened, then glucose, which is a source from the carbohydrate, it actually converted into the pyruvate, and this is happening by the process known as glycolysis. Right? So during the glycolysis, ATP is produced and NADH is produced. Right? So if there is no oxygen in the environment, then pyruvate is converted into different type of products under different type of fermentations. Right? For example, lactic acid, ethanol, acetic acid, propionic acid and butyric acid by different type of microorganisms, as you can see here, right? So for each type of fermentation, there are different type of microorganisms are responsible for that, right? Next one is the most important one, that is the alcoholic fermentation we discussed earlier. So this one is also occurs in the absence of oxygen, and in this case also glucose is converted into pyruvate molecules where NADH is produced, right, through the glycolysis process. right? And then acetaldehyde is produced under the anaerobic conditions, and this acetaldehyde is then further produces the ethanol by regenerating the NAD plus molecule, which is again used in the glycolysis for the production of ATP. Right? So this is the cycle for alcoholic fermentation, which occurs in the absence of oxygen. So this is all about the fermentation you should know about. It is a very important topic and very basic questions comes from it. So that's all for the today. Thank you.